final volume up to 15 milliliters with complete medium. Wash the cells by centrifugation and resuspend the pellet in 1 milliliter of fresh complete medium. Then, count the cells and adjust the volume to a concentration of at least 2 times 10 to the 4th NK cells per 100 microliters in complete medium. To stimulate the isolated NK cells, first gently mix by pipetting at least 5 times and aliquot 100 microliters of the cells into the appropriate wells of a 96 well D bottom plate. Switch off the light and add anti-CD107A antibody to each well of the NK cells. Then, carefully mix an aliquot of K562 tumor cells at a 2 times 10 to the 4th cells per 100 microliter volume by pipetting at least 5 times, and transfer 100 microliters of the tumor cells to each well planned for the stimulation.
Precise pipetting and the preparation of a 1-to-1 effector target ratio are of key importance for the accurate assessment of NK cell function.
Next, add IL-2 and IL-15 to the stimulation wells, and mix all of the wells by pipetting at least 5 times. Incubate the plate, protected from light, for 3 hours at 37 degrees Celsius and 5% carbon dioxide. After the first hour, with the light switched off, add freshly prepared protein transport inhibitor solution to each well with careful mixing, and return the plate to the incubator. At the end of incubation, carefully mix the co-cultures and transfer them into their corresponding flow cytometry tubes. Add 1 milliliter of wash buffer to each tube for centrifugation and resuspend the pellets in 99 microliters of PBS plus 1 microliter of fixable dead cell marker per sample. Mix the cells by vortexing, followed by a 15-minute incubation at room temperature in the dark. At the end of the incubation, wash the cells in 1 milliliter of wash buffer and resuspend the pellets in 84 microliters of wash buffer plus 16 microliters of the appropriate cell surface antibody cocktail. Mix the samples by vortexing and incubate for 20 minutes in the dark at 4 degrees Celsius. Then, wash the cells in 1 milliliter of wash buffer and resuspend the pellets in 100 microliters of a cold fixation solution. Mix by vortexing, followed by 10 minutes at 4 degrees Celsius in the dark. Now, wash the cells in 1 milliliter of wash buffer and resuspend the pellets in 90 microliters of permeabilization buffer. Stain with 10 microliters of the appropriate intracellular antibodies and mix by vortexing. After 30 minutes in the dark at 4 degrees Celsius, wash the cells 2 times in 1 milliliter of fresh permeabilization buffer. Then, resuspend the samples in 400 microliters of fresh permeabilization buffer, mix well by vortexing, and place the cells on ice until their analysis by flow cytometry. As soon as possible, transfer all tubes into the flow cytometer and start the data acquisition.
Đây là cái máy fax để mà nó sẽ đọc, như chúng ta biết, nó sẽ làm đọc một lần có thể lên tới 10.000 cell. Và từng cái cell nó sẽ có một cái máy hút để nó đưa là single cell chạy xuống cho một cái dòng chảy, cho nên nó gọi là flow cytometry ha.
Here, gating strategies for analyzing the degranulation and cytokine and chemokine production of the whole NK cell population, as well as 3 different NK cell subsets, are illustrated. Unstimulated NK cells from healthy donors produce neither interferon gamma nor MIP-1 beta and do not express CD107A on their surface.
Thông thường á, một cái kết quả FACS ha,
NK cells
nó sẽ chia rất là nhiều thứ. Ví dụ như các bạn, cái kết quả FACS thì mình phải mở ra rất nhiều cửa sổ, và mỗi cửa sổ nó sẽ là có 2 cái marker. Thì thông thường chúng ta sẽ thấy là có những cái marker nó sẽ là cho một số tế bào granule cell, là những tế bào có hạt. Thì NK cell nó thuộc cái nhóm tế bào có hạt ha. Và những cái tế bào mà mình sau khi mình kích ứng với interferon gamma chẳng hạn, kích ứng với MIP 1 beta, rồi sau đó những cái tế bào này mình sẽ là thu nhận ha, mình thu nhận những nhóm tế bào này và mình tiếp tục chạy thêm những cái marker để chứng minh rằng nó sẽ có những cái marker chuyên biệt cho NK cell, và những cái marker khác liên quan tới những tế bào mà nó giả định giống NK cell cũng phải được loại trừ. Ví dụ như vậy, thì từ những cái việc phân tích và chúng ta thấy là mỗi một cái chấm này, một cái chấm đây chính là những cái tế bào. Ở đây thì chúng ta thấy là tế bào rất là đồng nhất ha, tập trung dương tính với cái CD56 và âm tính với INF gamma. Hoặc là ở đây chúng ta sẽ thấy có nhiều cái nhóm tế bào, chia thành nhiều nhóm tế bào, trong đó có một nhóm tế bào nó cũng đồng nhất, nhưng mà những cái tế bào còn lại là nó heterozygous, tức là nó phân tán ra nhiều thứ, có thể là nó dương tính vào một cái CD56 chẳng hạn, nhưng những cái nhóm tế bào này nó lại âm tính với CD56 mà dương tính với CD16, hoặc cái nhóm tế bào ở trên cùng đây thì nó là dương tính với CD56 và dương tính với cả CD16. Thì từ đó chúng ta sẽ biết được là cái phần trăm tế bào NK cell thật sự mà trong cái mẫu người cho là bao nhiêu ha. Ví dụ ở đây nó chỉ có chiếm được 2.29% là tế bào mà đạt chuẩn để mình có thể là trữ lại để mà tiêm vào những cái nhóm bệnh nhân mà ung thư ha. Tế bào NK cell nó cực kỳ hiệu quả với việc nhận dạng cái tế bào ung thư.
Cytokine and chemokine production of the whole NK cell population, as well as 3 different NK cell subsets, are illustrated. Unstimulated NK cells from healthy donors produce neither interferon gamma nor MIP-1 beta and do not express CD107A on their surface. In contrast, NK cells stimulated with tumor cells and cytokines produce significant amounts of intracellular interferon gamma and MIP-1 beta, with over 20% of the cells demonstrating degranulation upon tumor cell interaction, as indicated by their CD107A cell surface expression.
Okay. Một video khác là chúng ta sẽ thu thập tế bào parenchyma và non-parenchyma của toàn bộ cái mẫu mô gan. Chúng ta biết là những cái bài trước chúng ta đã nói rồi, tức là một số cơ quan nó có khả năng là tái tạo lại chính cái cơ quan đó. Ví dụ khi một cái lá gan mình bị cắt đi, một tổn thương đi, cắt loại bỏ, thì từ trong cái lá gan đó nó cũng sẽ tái tạo lại các cái tế bào để định hình cái mẫu mô gan. Thì ở đây người ta thử nghiệm việc mà thu thập những cái tế bào stem cell mà có cái marker CD133. Đây là cái marker chính yếu của cái liver stem cell ha. Và cái cách thu nhận như thế nào chúng ta sẽ xem. Đây là cái mẫu gan của người.
This video demonstrates a procedure for isolating liver stem cells to study clonal expansion using CD133 cell surface expression following chronic liver injury.
Sorry, đây của trên mẫu động vật ha. Murine.
First, the liver is removed from a mouse with chronic liver injury and digested to produce a single-cell suspension. Red blood cells lysis buffer is then added to remove the red blood cells. And the solution is applied to a magnetic CD45 antibody-containing column to remove CD45 leukocytes. The CD45 depleted cells are then sorted to enrich CD133 positive.
Đây là cái màu cái nhóm tế bào mà người ta cần đạt được ha. Khoảng 15% tế bào gốc CD133 ha. Và nó nhuộm với PE và chạy qua cái máy fax.
Liver stem cells. Finally, isolated CD133 positive liver stem cells are cultured and expanded in vitro. Gene and protein expression analysis demonstrate that the isolated CD133 positive stem cells are able to differentiate into both hepatocytes and cholangiocytes.
The main advantage of this technique over existing methods, such as co-localization of markers using immunohistochemistry techniques, is that single cells can be isolated for clonal expansion and functional analysis to confirm stem and progenitor cell phenotypes. Demonstrating the technique will be myself and Hian Dang, a graduate student in my lab.
Prepare all buffers and media 24 hours in advance and store them at 4 degrees Celsius until use. All solutions, media, instruments, filters, and tubes should be sterile and handled with sterile technique to reduce the risk of contamination. Wipe the abdominal area of a euthanized mouse with 70% ethanol. Using sterile instruments, open the abdominal cavity and explant the liver en bloc. Remove the gallbladder from the explanted liver. Then transfer the whole liver to a laminar flow hood in a closed sterile dish. In a laminar flow hood, use a sterile razor blade to mince the liver with a combination of multiple horizontal and vertical cuts for 1 minute. Divide the minced liver into 4 parts and place each part in its own 15 milliliter tube with 10 milliliters of enzyme digestion solution. Then place the tubes in a 37 degrees Celsius water bath for 45 minutes in a shaker set to 1 to 2 cycles per second. Following the incubation, wipe tubes with 70% ethanol. Then, under a laminar flow hood, pipette the digested liver pulp through 70 micron mesh filter and collect the flow-through in a sterile dish. To remove the parenchymal fraction, which contains the larger hepatocytes, rinse the filter with 2 milliliter aliquots of sterile supplemented DMEM F12 medium. Use the rubber end of a syringe plunger to mash the digested pulp through the filter. Repeat this process 5 times to make total volume of approximately 20 milliliters. Divide the filtrate into 2 equal 15 milliliter tubes. Then centrifuge at 50 times G for 1 minute at 4 degrees Celsius. Following the spin, transfer the supernatant to a tube labeled supernatant number 1 and discard the parenchymal pellet. Next, to collect the non-parenchymal fraction, which contains the stem and progenitor cells, centrifuge supernatant number 1 at 50 times G for 1 minute. Transfer the supernatant to a tube labeled supernatant number 2 and again discard the pellet. Then centrifuge supernatant number 2 at 50 times G for 1 minute. Following the spin, transfer the supernatant to a tube labeled supernatant number 3 and discard the pellet. Finally, centrifuge final supernatant number 3 for 180 times G for 8 minutes to obtain non-parenchymal fraction. Following the spin, discard the supernatant and proceed immediately to the next step of the protocol, in which the red cells in the pellet are lysed. The red cell lysis buffer and Miltenyi buffer used for this part of the procedure should have been prepared the night before and stored at 4 degrees Celsius, according to the instructions in the accompanying written document. Working in a laminar flow hood with the cells and solutions on ice, Resuspend the non-parenchymal pellet from step above in 1 milliliter of 1x diluted red blood cell lysis buffer and transfer it to a 5 milliliter tube. Cap the tube and gently vortex it for 5 seconds. Incubate for 15 minutes at 4 degrees Celsius, protected from light. After 15 minutes have passed, centrifuge the tube at 200 times G for 5 minutes. Following the spin, discard the supernatant, which contains lysed red blood cells, and resuspend the pellet in 1 milliliter ice-cold and sterile Meltony buffer. Centrifuge at 200 times G for 5 minutes. After the spin, discard the supernatant and resuspend the pellet in 1 milliliter ice-cold and sterile Meltony buffer. Then remove 10 microliters of this cell suspension and add 10 microliters Trypan Blue. Count the non-parenchymal cells using a hemocytometer. To perform the CD45 hematopoietic cell depletion from the non-parenchymal fraction, begin in the laminar flow hood by adjusting the concentration of the cells to 10 to the 7th to 10 to the 8th total cells in 100 microliters of Meltony buffer. Be sure to keep the cells and buffers cold throughout the entire procedure. Next, apply 20 microliters of Miltenyi CD45 microbead antibody per 10 million cells and incubate at 4 degrees Celsius for 15 minutes. Following the incubation, add an additional 2 milliliters Miltenyi buffer and centrifuge at 200 times G for 5 minutes. After the spin, remove the supernatant and resuspend up to 10 to the 8th cells in 1 milliliter Miltenyi buffer. In a laminar flow hood, place a Miltenyi LD magnetic column in a magnetic holder. Place a sterile 5 milliliter tube below the filter to catch the filtrate. Then load 2 milliliters Meltony buffer to perform a filter pre-wash. Once the wash is complete, load the cells onto the column. Once the cell suspension is within the column, add 1 milliliter Meltony buffer to wash the column. Perform this wash 2 more times. Do not use the plunger provided with the column to increase the speed of filtration. Once all of the washes have been performed, centrifuge the collected filtrate of CD45 depleted non-parenchymal cells at 200 times G for 5 minutes. Discard the column with the retained CD45 positive cells. Next, flow cytometry is performed to isolate CD133 positive cells. Resuspend cells in Miltenyi buffer at 10 to the 7th cells per 100 microliters. Aliquot 100 microliters into 3 tubes. To the first tube, add 2 microliters of CD133 PE conjugated antibody. To the second tube, add IgG PE conjugated antibody as a control. The third tube of cells will serve as the unstained control. Incubate at 4 degrees Celsius for 15 minutes in the dark. Following the incubation, resuspend in 2 milliliters of staining buffer. Centrifuge at 200 times G for 5 minutes. Discard supernatant and resuspend the pellet in 1 milliliter Miltenyi buffer. Next, on the flow cytometer, use unstained cells and IgG PE stained cells to adjust the sorting parameters for optimized gating of CD133 positive cell population. R-phycoerythrin, or RPE, can generally be used with any flow cytometer that has a laser that emits 488 nanometers. Finally, isolate the CD133 positive cell population using CD133 positive gate and collect the cells in sterile filtered oval cell medium. The cells can now be cultured and clonal isolation can be performed, followed by real-time PCR to assess bipotential status. CD133 positive liver stem cells were isolated from wild-type and MAT1A knockout mice, as described in this video. As seen here, CD133 positive cells make up 0.4% of the highly enriched CD45 depleted non-parenchymal control fraction derived from uninjured liver from wild-type mice. In the genetic knockout MAT1A minus, which is a model of chronic liver injury, the CD133 positive population expands tenfold or more in the same highly enriched fraction. Following sorting, cells were cultured and clones were expanded. Shown here are phase contrast images of 4 colonies derived from single CD133 positive cells expanded on laminin-coated 96-well plates. These cells are 7 days post-isolation